some pep to the mini prep by knowing what goes on at each and every step. So a mini prep is an alkaline lysis kit, and it's a way that we can isolate a plasmid, so a circular piece of DNA that we stuck into bacteria, from the bacteria's own DNA. So bacteria are really helpful in that we can stick these plasmids inside of them and they'll make lots of copies from them. If we want to purify out those copies, we want to purify out the plasmid, we're going to need to break open the bacterial cells or lyse them and then isolate that plasmid. And a mini prep is a convenient way to do this. Now, it's so common in biochemistry and molecular biology that it's used all the time by tons and tons of labs. And so if you go into a lab, you'll see lots of these boxes or similar boxes. And so inside of this boxes, basically, it's just like a commercialized version of an alkaline lysis kit, and it uses columns which make things more easy to like purify out the DNA after you've isolated it. But it can also hide the cool biochemistry that's going on. So today, let's talk about what's going on in each step. Not only is it cool, but it can also help you if you need to troubleshoot or things like that. I'm going to be talking in terms of the Keogen kit, but it's a pretty standard kit a lot of labs use, but you can also use it with like homemade buffers, and I will post a link to recipes for those. The idea with all of these, this alkaline lysis method, is that alkaline, this is going to be like basic, as opposed to acidic, and lysis, you're going to be breaking open cells. And so we're going to be breaking open bacterial cells in order to isolate the plasmid. And we're going to use alkaline conditions in order to kind of unzip the DNA. So we're going to break open the cells and we're going to unzip the DNA. And when we unzip the DNA, well, why are we doing this? We're doing this because it's going to let us selectively purify the plasmid as opposed to all of the bacterial DNA, the genomic DNA, the gDNA. And so this plasmid is going to be smaller than that bacterial DNA. It's like the smaller circular thing, about like 2,000 base pairs, maybe at the most. Whereas with the E. coli genome, then you're talking about like 4.7 million or something. And so we have a lot, that's going to be a lot bigger. And so if you were to unzip the strands and then tell them to rezip, it would be harder for that bacterial DNA, the gDNA, in order to zip than it would be for the plasmid DNA. And so we're going to take advantage of this to selectively purify out the plasmid. And then we're going to use a variety of different steps to kind of wash it off with these little columns. Now you can do these column washing steps in like a centrifuge typically, but if you have a lot of samples, you can also use a vacuum manifold. And so let's go into a little more detail about the procedure. So first, you're going to have to grow up a lot of the cells. And so we've taken their plasmid and we stuck it into bacterial cells. And then we let them, so that step is called transformation. And then we plated it on like an LB agar plate with antibiotics so that we're selecting so that only cells that have the plasmid are able to grow, because that plasmid has the antibiotic resistance gene. And so the bacteria that have the plasmid, they're going to grow, and they're going to grow in these little clumpy dots called colonies. Basically, the bacteria, they kind of take root and then they grow on top of each other. And so you get these clumps called colonies. And then what you're going to do is you're going to actually need more of that than just that. So you're going to do a liquid culture growth overnight with like a 5 ml culture of lysogeny broth, so like LB. And so you just take a colony, you kind of take your toothpick or your pipette tip and you take it, you like flick the little colony a little, get a little bit of that goop on there, drop it into your LB with antibiotic and stick it on the shaker overnight. And then you come back the next morning and it should have grown. And so you get this nice cloudy mixture. And now what you need to do is you need to spin it down and then resuspend it. So basically, we want to remove that media, the food that it was growing in. And so in order to do that, we just spin it down so that the cells go to the bottom and then the media is above it. And we take off this media, we can just like pour it off into some bleach. And now we need to resuspend the pellet because we want it to be in liquid now, but we want it to be in clean liquid, and we want it to be in liquid that we define, and not whatever the bacteria, whatever was in that media that maybe the bacteria was secreting and that sort of thing. And so we're going to now, we've spun it down, in a benchtop centrifuge like this. And now we are going to pour off that media and resuspend it. And so let's talk about the resuspension buffer. Basically, this resuspension buffer, when we talk about a buffer, we often use this term buffer kind of like just as a generic word for a solution. But buffer here really is a buffer. It's a pH buffer. And so it basically is going to buffer or kind of like even out changes in pH so it is able to maintain a stable pH. And the buffering agent that's in here is tris. And so tris is just a common buffer, and it's set to keep pH about 8. We're going to later introduce the alkaline agent, so make it so that it's higher than 8. But for now, we want to keep things stable, because at this point, we don't want the cells to break open yet. Our point is just to resuspend them in some more liquid, and then we're going to work with them. And so right now, we want to keep them from breaking open. And so to help us keep them from breaking open, we're also going to have glycerol. And so glycerol, what it's going to do is it's going to help with the osmotic balance. And it's going to keep the cells from bursting for now. So basically what can happen is that you add, if the solution outside of the cells is too dilute compared to the stuff inside of the cells. Now what's going to happen is the stuff in the liquid from outside the cells is going to flow into the cells. You can think of it kind of like trying to even things out. It's not really what's going on, but this liquid is then able to enter the cells, and this could cause the cells to like swell up and even burst. By adding glycerol, what you're doing is you're just kind of adding stuff outside of the cells. And so now there's more stuff outside of the cells, and you have less of that osmotic pressure imbalance and stuff. And so you have less of the liquid coming in, and this is going to keep the cells from bursting. What else do we have in here? We actually have RNase A. Normally in the lab, we're trying to stay away from RNase A at all costs. And so RNase A is an RNA degrader, and it's just like ubiquitous. It's like everywhere. And it's, when you're trying to work with RNA, it's like a huge pain, and you take a lot of care in order to avoid it, because it's really hard to get rid of, and it's really like sturdy and stuff. So basically here we want to add it though, because we want to get rid of the RNA. We just want the DNA, and so we can add RNase A. Note that because we add RNase A, so when it comes in the kit, typically you have to add the RNase A, and then once you add the RNase A, you keep it in the fridge. And so the other components you keep at room temperature, but this buffer P1 is going to be in the fridge once you add the RNase A. And so the reason why RNase A is not going to degrade the DNA is because DNA is safe because it doesn't have an OH. So RNase A is actually this, it's pretty cool. And well, not when you don't want your RNA being chewed up, but what it does is it kind of like uses water to get the RNA to attack itself. And so RNA differs from DNA in that, well, in one, it has a U instead of a T, but it also has a 2' OH. And this 2' OH is then able to kind of attack the phosphate group between the RNA letters with the help of RNase A, kind of egging it on. And so but DNA doesn't have that 2' OH, so DNA will be safe. So we can add this RNase and get rid of the RNA with keeping our DNA. And finally, we have one more thing. We have thymolphthalein. And this is this like lyse blue reagent. And it's like, you don't have to have it. You don't have to add it in. But it's going to serve as a pH indicator if you do. At high pH, and so remember high pH, so high pH, we're talking about basic or alkaline conditions, which is what we want later when we're going to unzip the DNA. And so right now we have tris to keep things at around 8, but later we're going to want to raise things. And we want to make sure that we actually have raised things and that we've raised things evenly throughout the mixture. We want to make sure that we've mixed things adequately. And so this thymolphthalein, basically at about pH of 10, it's going to turn blue. And then later, when we want to neutralize the solution, it'll turn back to being colorless. And so this is going to help us make sure that we mix things thoroughly and that sort of thing. So now it's time to lyse the cells. Now with lysis, basically, what do we have to deal with? We have to deal with this bacterial cell, which is going to have this membrane. It's like double membrane, and it's going to have phospholipids like our cells do, and it's going to also have these lipopolysaccharides, or these like LPS. And so basically these phospholipids and this lipopolysaccharides, at least part of it is going to be it's going to be hydrophobic, so water avoiding. And so these water avoiding parts are kind of stuck inward. And then they have these negatively charged parts on the outside. And what can happen is that normally these bacterial cells, so because they have all that negative parts on the outside, those negative parts would repel one another because like charges repel. Well, what's going to happen is that normally they have magnesium and calcium and ions, kind of these charged particles, these cations. Their positive charge is kind of balancing out the negative charge and keeping this membrane happy. What you can do is, if we want to destabilize the membrane, we can add EDTA. And so EDTA is a chelator. Basically, it bites down around metals, and it's going to help steal those balancing metals so that you still have a negative charge, and now those negative charges are going to start repelling each other, and you still have this kind of like hydrophobic part. What you can do is you can actually add a soap or a detergent. So in this case, the detergent is an artificial soap called SDS, sodium dodecyl sulfate. If that name sounds familiar to you, that's what we use with SDS page to separate proteins by their size by sending them through a gel. The SDS is going to be used to denature or like unfold the proteins, keep them soluble, and give them a negative charge to send them through the gel. And so in this bacterial membrane, there are proteins, and there's also going to be those like hydrophobic things. And so both of these are perfect for getting broken up by SDS. And so in this way, the SDS is kind of able to sneak its way into the membrane, help break things up, and denature the proteins that are embedded in the membrane. Because although we often think of this membrane as being this like phospholipid thing, which it is, it actually has a lot of it is actually proteins. And so this SDS can help with this. Okay, so now we've broken open the cells, and that's not all that's in the lysis buffer. What's also in the lysis buffer is going to be the alkaline parts. We're going to have the NaOH. Finally, we get that base. And what this is going to do is basically what it's going to do is it's going to disrupt the hydrogen bonding. And so the reason why strands of DNA stay together is through hydrogen bonding with the type of partial charge attraction. It's kind of like a stronger type than your just average attraction, but it's not a strong bond like the bonds that are actually holding the letters together within the DNA chain. But it can hold the chains of DNA to one another, zip them up. And it takes advantage of, relies on having a hydrogen on one strand and on one base, and then like an oxygen on the other base, or a nitrogen. And so this hydrogen serves as what we call a donor, and then this other one serves as an acceptor. And we get this attraction. Much more on this in other posts. But what can happen is that at a high pH, those NHs can lose their H's. And so you lose that donor. And if you lose that donor, well, now this is going to disrupt the base pairing. And if you disrupt the base pairing, well, now those strands are going to come apart. And note that you would not want to go to a high pH with RNA, because what can happen is that you get hydrolytic cleavage, where basically, similarly to how we had RNAs get the RNA to attack itself by activating water, here you actually have basically the hydroxide ion. Basically, it can do the same thing, and so you don't have to activate the water because you already have this base here that can steal that H from the 2' OH, get the oxygen to attack the phosphate. So bottom line is that the RNA can attack itself under high pH conditions and degrade, but you don't need to worry about this with the DNA. So we're able to increase the pH, take it to these basic or alkaline conditions where we can denature the strands of the DNA. So when we denature the strand, remember that if you added that lyse blue, that thymolphthalein, things should turn blue, and you want things to turn blue throughout the mixture. And so basically, this will help you see that you've thoroughly mixed it. And when you're mixing it, you're just going to want to do it by inverting, like inverting the tube over and over. I mean, not over and over and over, but I mean like several times until you get that nice blue throughout. And the thymolphthalein is going to help you know when you've done that. You want to be careful when you're doing this because although we didn't have to worry about the NaOH causing the DNA to like attack itself and break, we do need to worry about breaking the DNA by being too harsh with our mixing. So you don't want to stick this on a vortex or anything. Instead, just invert the tube several times in order to thoroughly mix it. You let that go for a few minutes. Let things lyse. Oh, sorry. Yeah, let things lyse. Let the DNA break apart. But we don't want to let it go forever. We need to neutralize things. And so to neutralize a base, well, you add an acid. And so in this case, we're going to add potassium acetate. Now, potassium acetate, basically, this is a carboxylic acid group. And so when it deprotonates, you get a carboxylate. And so this is where we get acetate. And so this is basically a carboxylate group, and it's negatively charged. And so now we need a counter ion. We have something that hangs out with it. We have to give us a salt. We have potassium acetate. And so when you have potassium acetate, when you stick it in water, now what's going to happen is that potassium is going to come off. And so you're left with this acetate group and potassium. And now this potassium, I don't know if you've ever had a potassium chloride buffer and then you try to prepare an SDS page sample and you end up with things getting precipitated. This is because potassium dodecyl sulfate is not soluble. And so, but sodium dodecyl sulfate is, and so this is why we can use SDS. But if you have SDS and then you add it to your mixture, which has potassium in it, well, now this can cause things to precipitate. And just if you're trying to do a sample with SDS where you have potassium in the sample, it's good to like heat your sample up right before you load it to prevent the precipitation. But now we want to take advantage of the precipitation because the precipitation is going to help us isolate out or take away the genomic DNA, that gDNA. So once we've unzipped those strands, basically that gDNA is going to get tangled up with the SDS-coded proteins and lipids and all of this stuff. And thanks to the KDS being insoluble, this is all going to kind of just like clump out. And it's going to like, so it's going to pull all the associated stuff with it, and you kind of get this white fluffy stuff. And it's also going to smell kind of vinegary because of this potassium acetate, and this is like acetic acid. This is like vinegar, so you get this like vinegary smell. And what you're doing is you're taking away the gDNA and all the proteins and all the lipids and all of that stuff. But your plasmid, because it's smaller, it's going to rezip again because now we're back to neutral, where those bases can get their protons back, and they can pair back up because they can find one another. They're just like these smaller circular pieces. It's more easy to find one another to rezip than this big gDNA. Also in the neutralization buffer is going to be guanidium chloride. And so this is going to help it bind to the column. And our next step is going to be to go onto that column. But before we go onto that column, first we actually have to spin things down. Because basically, after we do that lysis, after we do the neutralization, well, now we're going to have all this clumpy stuff throughout our mixture. So it's going to be kind of like white and cloudy and stuff throughout the mixture. And now we need to spin it down to actually get rid to isolate that stuff. And so at this point, we're not going on the column yet. At this point, what we're doing is we're doing this spin down in just a normal tube. We do all of this in just like a normal Eppendorf tube. And then once we actually do the spin down, now we can take the supernatant, so take the liquid, so which has our plasmid, because our plasmid is soluble, but all the other stuff, that gunk has been pelleted out. We take that liquid supernatant and now we can apply it to our column. And this column for now is going to go into one of these waste tubes. At the end, when we're ready to elute, so when we're ready to get the DNA off, then we're going to stick it into a clean cap tube. But for now, we're going to use this capless waste tube. And now basically the protocol is similar to whenever you do like a PCR purification or any other sort of like DNA spin column. So we had added the guanidinium chloride, and the guanidinium chloride is going to be, it's a chiotropic salt, so it's going to kind of like help break up the water coat. What we want to do is we want our DNA to stick to the membrane on this column. And so we want to disrupt the water around the membrane. And then we're going to make it the conditions so that it will bind to the membrane, and then wash everything else off, and then change the conditions so that the DNA will come off. And so basically the DNA binding conditions, so it's like buffer PB, this like binding buffer. Basically it has an acidic pH. So basically at this acidic pH, there's going to be a lot of H's, a lot of protons. And this membrane is a silica membrane. And the silica membrane, basically when it's protonated, it'll be neutral, but when it's deprotonated, it'll be negatively charged. And DNA is a negatively charged backbone. So in order for the DNA to bind to the silica membrane, you don't want the silica membrane to be negatively charged. And then we have the guanidinium chloride. Well, guanidinium chloride is basically it's going to loosen up the water coat around the DNA. It's also going to help denature any proteins, although we shouldn't have any of those left. And it's going to provide a positive that's going to neutralize the negative of the DNA backbone. And this is going to allow it then to be less soluble and to bind, make these more hydrophobic interactions and things with the silica membrane. And so under these conditions, the DNA will bind. In addition, you have isopropanol, which is basically going to help them find each other and kick off the water. And so you're basically breaking up the water coat, and you're kind of making it so that the water network is easier to see through. And so these molecules can find one another. Now what's going to happen is you're basically have things on the column and you want to wash. And so you have like this buffer PE. And basically we've gotten rid of the proteins and all that big stuff before, but now we have a bunch of excess salts. And so we want to wash it off. And so ethanol is going to dissolve the salts, but not the DNA. And so this is going to allow us to wash the salts off. But when you do this ethanol wash, the ethanol can actually interfere with later steps. And so you want to make sure that you spin it twice to remove excess. So you spin, and then you don't even add anything more, but then you spin it again just to make sure all the ethanol really got through. And typically this buffer, you have to actually add the ethanol. It comes without the ethanol, and then you add the ethanol to it, and then you mark on the top of the bottle that you've added it so that people know that you've added it. Now, once you've washed everything off, now finally it's time to get our DNA to come off. And so we need to elute it or get it to come off. You can do this in the elution buffer that it comes with, or you can do it in water. Basically, you want to reverse the conditions that got it to bind. So you want to go to a higher pH where that silica membrane is going to start losing protons. It's becoming negatively charged, and DNA's backbone is negatively charged, so now they start repelling. And the DNA you also have lower salts, so basically the water molecules are able to form see the DNA better and things like this. And so the DNA is going to dissolve, and when it dissolves, now it can come through the column. And so the reason why it was getting stuck in the membrane before wasn't that it was too big to get through membrane pores or something. It's just that it was physically sticking to the membrane. And so now you've eluted off your DNA, you've eluted off your plasmid, and you want to see what the purity is and what your yield is, like what's the concentration. The concentration, if you want it more concentrated, you can elute in less liquid. Typically when you do the water elution, you basically add the water, let the water sit on the membrane for a minute, and then spin it to take it out. And remember, at this point, you want to be in your clean tube, not in the waste tube. And then you can go over to like a spectrophotometer, like a nanodrop. What it's going to do is it's going to shine light through your sample, and the DNA in the sample is going to absorb UV light. And based on how much light got absorbed, you can then figure out the concentration. And by looking at the absorbance at different wavelengths, you can figure out the purity. And so much more on that in other posts. But at the end of the day, you hopefully have a nice pure plasmid. You can now go stick that plasmid into your freezer and send it off for sequencing or do whatever you want to do with it. But you've taken advantage of the ability of this alkaline lysis method to selectively purify out that plasmid from the genomic DNA, from the lipids and all that gunk. So I hope that helps you understand what's going on in a mini prep. So basically you take your colony, you grow up a lot more of it, then you isolate those cells. Once you've isolated those cells, you break them open. And so you break them open using SDS as well as NaOH. So NaOH, this is where we get our alkaline part. This is our base. It's going to raise the pH, unzip the DNA. And then once you unzip the DNA, you can neutralize things. So you add acid to bring the pH back down, allow the DNA to rezip, but the genomic DNA, the bacterial's own DNA, it's too big to rezip easily. It's going to get tangled up in this like potassium diacetyl sulfate mixture of like proteins and all sorts of gunk. And it's going to get pulled down when you centrifuge things, but your plasmid is going to have been able to rezip. And so now you can take that liquid above the supername. So after you spin it, you spin out all the gunk. You take the liquid, and now you can use a spin column in order to purify it, get the DNA to bind to the column, wash everything else off, and then get your DNA to come off or elute all by itself. And that is how a mini prep works. And happy prepping.